A cell is the basic structural, functional, and biological unit of all living organisms. It is the smallest unit of life that can carry out all the essential processes of life. 1. Basic unit of life. All living organisms are made up of one or more cells. 3. Cell membrane. Controls entry and exit of substances. 4. Cytoplasm. Jelly-like substance where cell activities occur. 5. Nucleus. Control center of the cell in eukaryotes. 6. Organelles. Specialized structures like mitochondria, ribosomes, etc., perform specific functions. 7. DNA. Genetic material present in all cells. 8. Reproduction. Cells divide to form new cells, mitosis or meiosis. Cytoplasm. 1. Cytoplasm is the jelly-like substance inside the cell, between the cell membrane and the nucleus. 2. It is made up of cytosol, fluid part organelles like mitochondria, ribosomes and inclusions, stored nutrients or pigments. 3. Cytoplasm supports and holds the organelles in place and allows them to function properly. 4. It helps in transporting materials within the cell and is the site for many chemical reactions, like glycolysis energy production. 5. Cytoplasm also helps the cell maintain its shape and structure through a framework called the cytoskeleton. 6. It plays a key role in cell movement, growth, and divisions. Composition of the nucleus. The nucleus is composed of several key components. Nuclear envelope membrane, a double lipid bilayer membrane that surrounds the nucleus and separates its contents from the cytoplasm. Nucleoplasm, nuclear sap, a gel-like substance that fills the interior of the nucleus, rich in enzymes, ions, and nucleotides. Nucleolus, a dense, spherical region within the nucleus responsible for ribosomal RNA, rRNA, synthesis and ribosome subunit assembly. Structure of the nucleus. Double membrane envelope. Outer membrane is continuous with the rough endoplasmic reticulum, RER, and may have ribosomes. Nuclear pores. 1. Present in the nuclear envelope, which surrounds the nucleus. 2. Allow exchange of materials between nucleus and cytoplasm. 3. Made of proteins forming a complex called the nuclear pore complex, NPC. 4. Control movement of RNA, ribosomal subunits, proteins, and ions. 5. Selective transport, only specific molecules can pass through. 6. Large molecules like mRNA require active transport. 7. Found in eukaryotic cells only. 8. Each cell nucleus may have thousands of nuclear pores. Chromosomes. 1. Chromosomes are thread-like structures found in the nucleus of cells. 2. They are made up of DNA and proteins and carry genetic information. 3. Humans have 46 chromosomes in each body cell, 23 pairs. 4. They control hereditary traits like eye color, height, and blood group. 5. Chromosomes are visible only during cell division. 6. One pair of chromosomes determines sex, XX female, XY equals male. 7. Abnormalities in chromosome number or structure can cause genetic disorders. Smooth endoplasmic reticulum, SER. 1. Structure. A network of tubules and vesicles without ribosomes, making its surface appear smooth under a microscope. 2. Lipid synthesis. SER is responsible for making lipids like phospholipids for cell membranes, cholesterol, and steroid hormones like testosterone and estrogen. 3. Detoxification. especially active in liver cells. SER helps detoxify drugs, alcohol, and metabolic waste products. 4. Calcium storage. In muscle cells, SER is called the sarcoplasmic reticulum and it stores calcium ions required for muscle contraction. 5. Carbohydrate metabolism. It helps in converting glycogen to glucose in liver cells. Rough endoplasmic reticulum RER. 1. Structure. Consists of flattened sacs covered with ribosomes on the outer surface, giving it a rough appearance. 2. Protein synthesis. The ribosomes on RER synthesize proteins, especially those that are secreted from the cell like enzymes and hormones. 3. Protein folding and modification. Inside the RER, proteins are folded into their proper shape and often modified, e.g., addition of sugar chains. 4. Transport to Golgi. Proteins made in the RER are packaged in vesicles and sent to the Golgi apparatus for further processing and sorting. 5. Abundant in specific cells. RER is especially abundant in secretory cells, such as pancreatic cells, digestive ends. Lysosomes are membrane-bound organelles in animal cells that act as the cell's waste disposal system. Function: they digest damaged organelles, cell debris, and foreign substances using powerful enzymes. Structure: surrounded by a single membrane and contain acidic enzymes that work best at low pH. Formation: enzymes are made in the rough RER, processed in the Golgi apparatus, and sent to lysosomes. Types of digestion: autophagy, self-cleaning, and heterophagy, digesting external material. Diseases: defects in lysosomes cause storage diseases like Tay-Sachs and Gaucher's disease. Immune role: help white blood cells destroy bacteria and virus. Ribosomes are small, non-membranous organelles responsible for protein synthesis in all living cell. Structure: made of ribosomal RNA, rRNA, and proteins. Composed of two subunits. Large subunit joins amino acids. Small subunit read mRNA. Types: free ribosomes, float in cytoplasm, make proteins used within the cell. Bound ribosomes, attached to rough endoplasmic reticulum, RER, make proteins for export or membranes. Function: translate mRNA into polypeptide chains proteins using tRNA. Play a central role in gene expression. Found in both prokaryotic 70S and eukaryotic 80S cells. Also present in mitochondria and chloroplasts as 70S ribosomes. Plasma membrane, cell membrane. The plasma membrane is the outermost boundary of animal cells and inner to the cell wall in plant cells. It is made up of a phospholipid bilayer with embedded proteins, cholesterol, and carbohydrates. It is selectively permeable, meaning it controls what enters and exits the cell, like oxygen, nutrients, and waste. Functions include protection and structural support, cell communication and signal reception, transport of materials via passive diffusion, osmosis, and active pumps, endocytosis mechanisms. The fluid mosaic model describes its dynamic nature. Proteins float within the lipid bilayer like boats in a sea. Mitochondria, the powerhouse of the cell. Mitochondria are double membrane-bound organelles found in most eukaryotic cells. Their main function is to produce energy, ATP, through a process called cellular respiration. The outer membrane is smooth, while the inner membrane is folded into structures called cristae to increase surface area. Inside the inner membrane is a fluid-filled space called the matrix, which contains enzymes, DNA, and ribosomes. Mitochondria have their own DNA and ribosomes, allowing them to make some of their own proteins, suggesting an evolutionary origin from ancient bacteria endosymbiotic theory. They are especially abundant in active cells like muscle and nerve cell. Centrioles. Centrioles are cylindrical, microtubule-based structures present in most animal cells. Each centriole is composed of 9 triplets of microtubules arranged in a circular pattern, forming a 9x3 structure. These microtubules are tightly organized to provide structural stability. Centrioles always occur in pairs, positioned at right angles perpendicular to each other, and together they form a region called the centrosome. The centrosome acts as the microtubule organizing center (MTOC) of the cell. The primary function of centrioles is during cell division. Before mitosis or meiosis begins, centrioles duplicate, and each pair migrates to opposite poles of the cell. Golgi complex, the cell's packaging center. The Golgi complex, also called the Golgi apparatus or Golgi body, is a membrane-bound organelle found in eukaryotic cells. It consists of a series of flattened, stacked sacs called cisternae. It functions as the processing, packaging, and distribution center of the cell. Proteins and lipids synthesized in the endoplasmic reticulum are transported to the Golgi complex, where they are modified, e.g., by adding sugars or phosphate groups, sorted, and then packed into vesicles for delivery to their final destinations, either inside or outside the cell. The Golgi complex has two main faces. Cis face, the receiving side facing the Vacuole, storage and support structure. A vacuole is a membrane-bound organelle found in both plant and animal cells, but it is much larger in plant cells. Vacuoles serve various functions depending on the cell type. In plant cells, the central vacuole is large and filled with cell sap, a mixture of water, nutrients, and waste products. It plays a major role in maintaining turgor pressure, which helps the plant stay rigid, and storage of nutrients and waste products. In animal cells, vacuoles are smaller and involved in processes like digestion, storage, and waste removal. Vacuoles help in maintaining cellular homeostasis by regulating water balance and acting as a storage site for chemica. Cytoskeleton, the cell's endoskeleton. The cytoskeleton is a dynamic, protein-based framework found in the cytoplasm of eukaryotic cells. It acts like an internal endoskeleton, giving the cell shape, strength, and flexibility. It is made up of three main types of protein filaments. 1. Microfilaments, actin filaments, support the cell's shape and help in movement. 2. Intermediate filaments, provide mechanical strength. 3. Microtubules, maintain cell shape, form the spindle fibers during cell division, and guide movement of organelles. The cytoskeleton also helps in intracellular transport, cell division, and movement of the entire cell, as seen in amoeboid movement. We will talk more about their functions and study the details more deeply. If you like the video, please like it, subscribe, press the bell icon, and thank you very much for watching the video till the end.